I'd like to thank the moderators, Drs. Kretzler and Barasoni, for the opportunity to speak with you today about the Kidney Precision Medicine Project, and specifically discuss with you our strategies within the Kidney Precision Medicine Project, or KPMP, to safely obtain protocol research kidney biopsies in a multi-center setting. And I'll start with 10 years back, where we were 10 years ago. This is Margaret Hamburg. At that time, she was the commissioner of the FDA, writing this piece in the New England Journal of Medicine, one of the leaders, visionary leaders in American medicine. And she used very strong language saying that we are failing as a scientific community and as a nation to adequately deliver the promise of science to diagnose, treat, prevent, or cure disease. We must invest in new methods, assays, standards, and models that will help speed the development, review, and approval of medical products that patients need and rely on. And certainly for us in the kidney field, our patients need us to develop new methods and models and standards and come up eventually with new types of therapies to make life better for people living with kidney disease. If we fast forward almost 10 years from those comments from Margaret Hamburg, the Kidney Precision Medicine Project was envisioned and initiated by the NIH and specifically by the NIDDK. And the rationale for this project is that the way that we describe our most prevalent kidney conditions for patients, acute kidney injury and chronic kidney disease, these are currently defined by the serum creatinine, the amount of urinary protein excretion, and in the case of AKI, by the amount of urine volume. And these measures, which we've used to define AKI and CKD as syndromes for the last 20-odd years, do provide important epidemiologic information and useful prognostic information, but they fail to cluster patients into specific disease mechanisms. And partially as a consequence, it's been difficult over the last several decades to come up with new pharmacologic therapies for the heterogeneous presentations of individuals with acute kidney injury or chronic kidney disease. And these efforts have been further limited by the failure of animal models to replicate human disease phenotypes, by the lack of recognition sometimes of the heterogeneity of individual people who experience AKI or have chronic kidney disease. And as a consequence, we haven't had the ability to identify and prioritize high-value human targets. And we haven't had the kind of tissue available to us from kidney biopsies to really understand on a molecular level what's going on in these syndromes in the kidney. Unlike fields like cancer, where biopsies are routine and progress has been remarkable over the last several decades. This is not a new concept. Adalbert Bolle was a German nephropathologist who for decades back in the previous century, in the 20th century, repetitively described how when we look at kidney biopsies, we see information that's not attainable simply by looking at the creatinine clearance or the estimated GFR or even the amount of albuminuria. And here you see from one of his publications, glomeruli from two individuals with identical GFR or creatinine clearance, one with a perfectly normal glomerulus and the other severely diseased. So there's much going on beneath the surface in the kidney that we don't always appreciate by our commonly used measures to classify kidney diseases. And as a consequence, KPMP was initiated now about 3 years ago, and the goals of KPMP are pretty straightforward: to identify new treatments for kidney diseases, to ethically collect kidney biopsy tissues from participants with acute kidney injury or chronic kidney disease, and I should add safely, ethically and safely collect biopsy tissues, to use those biopsy tissues to create a kidney tissue atlas, which we'll describe in more detail, and both Mike Eden and Laura Mariani will discuss in more detail in subsequent talks, and use these types of data that will emerge to define disease subgroups. And from the beginning of KPMP, we felt that patient engagement was at the core, has to be at the core of our activities. And as a consequence, as I'll show, patients have been involved in every aspect of what we do inside the Kidney Precision Medicine Project. So what do we mean by creating an atlas? Well, there's an older definition of an atlas, going back 500 odd years, if you will, as a collection of maps that tells you where you are on the planet. A more modern view of that would be to focus that collection of images, charts, and tables to illustrate a particular topic. For example, the Netter Atlas of Human Anatomy. But the kind of atlas that we're talking about for the Kidney Precision Medicine Project is quite different. We describe it as a dynamic, living visual representation of multiple data domains linked to spatial information inside the kidney. Think of it as the difference between, if you're of my generation, the Rand McNally Atlas, if you were driving from point A to point B, and you would turn the pages as you moved along the road to find out where to go next on your journey, and compare that to not just global positioning system, but imagine software like Waze, where not only do you not just go from point A to point B, but you get continuously updated information on what's the fastest way to get from point A to point B. Now, I mentioned that we view patients as equitable partners in KPMP, and we formed a community engagement committee, largely whose membership is largely patients from the very beginning of KPMP. And our patient participants are involved in all our meetings and calls. They are represented on all our committees, including representation and voting representation on the steering committee for the study. Our patients developed the first draft of the informed consent form for this study, which was then reviewed by the single IRB for the study. They've developed patient-facing materials, and they've had enormous influence on the policies and plans that we've adopted for KPMP, above and beyond the consent process, but also dealing with no-fault harm, insurance coverage, and return of results as well. I'm proud to say of the first 10 papers, publications that have come from KPMP, 3 of the papers have been written by our patient participants in the Community Engagement Committee. Shows the level of involvement that patients have in everything that we're doing in KPMP. Now, when we talk to both participants in the study and our Community Engagement Committee participants, what patients want to know is, what do I have? What will happen to me? What can I do about it? And what does it mean for my family? And these are the targets for the Kidney Precision Medicine Project. And we are taking a precision medicine approach that says, we're not going to hopefully group everybody or lump everybody together in the future under terms like CKD or AKI, but we'll know based on molecular and other information, what's the right intervention for the right patient at the right point in time. From the get-go, patients that were involved expressed concerns about the ethics of the study, the safety of the study, the costs of the study, and really are there to protect participants in the study. The ethics that are involved, it's understandable that people will be taking risk if they're undergoing a kidney biopsy predominantly for research purposes. That risk will be altruistic in nature, as it's not clear that there will be individual benefit to participants. That risk to be ethical has to be reasonable in terms of the amount of information that can be obtained and the kinds of benefits that will come to the public health and to society. To be ethical, the study has to absolutely focus on safety and minimizing discomfort and risk for participants in the study, and costs really have to be borne by the study and not by participants. These were the mandates that came from our patient participants in the study. A key question was so-called indemnification if harm does unfortunately happen to a participant. And a quote from one of the members of our Community Engagement Committee really resonated with us when he said, "It sounds unconscionable to put all the financial risk on the volunteer altruistic kidney tissue donor." This is not the norm in the informed consents for many studies, including NIH-funded studies, where participants, if they are harmed, do bear potentially some financial risk. So we took this to heart, and we made a concerted effort, and were able to obtain gift funds from professional society that allowed us to purchase no-fault harm insurance to ensure that any participant in the study would not suffer any financial harm from participation in the study. We also undertook a review of kidney biopsy-related complications, predominantly bleeding complications, in a structured way, and these data are being published in Kidney International. We've done everything that we can by surveying all of the known risks to mitigate and minimize those risks for participants in KPMP. We think holistically about ensuring participant safety pre-biopsy, during the procedure, and post-biopsy, and have systematically done everything that we can to minimize the chances of any kind of adverse event happening to participants as a consequence of the biopsy. This goes from making sure that only approved biopsy operators perform these biopsies, highly experienced biopsy operators with an excellent track record for safety. We've installed checklists prior to the procedure to make sure that all safety aspects are being accounted for, with prospective surveillance of any potential biopsy-related adverse events, surveys after the fact as well, and of course review of all safety data, both by a safety and adjudication committee and by a data safety monitoring committee. This is just an example of the biopsy safety checklist, which is reviewed prior to the biopsy to once again make sure that all safety factors have been taken into account prior to the biopsy. And we developed exclusion criteria based on bleeding risk for the study that makes the study conservative and minimizes the chances that there would be pre-biopsy any excess bleeding risk based on either the use of antiplatelet agents or any bleeding diathesis or out of control blood pressure as examples of exclusion criteria for the study. Now, as mentioned, the rationale for our participation in the study is altruistic. We do not believe that we can tell our participants that they will personally benefit from the knowledge gained in the study. It's possible, but really participation in the study means taking on some personal risk for the greater good. And the rationale for participation is simply that this is kidney diseases, CKD and AKI. These syndromes are a major public health problem. 72% of patients on dialysis have kidney disease that's been attributed to hypertension or diabetes. They're disproportionately African American. They may carry genetic risks. The standard of care often does not include a kidney biopsy. In episodes of acute kidney injury, which are highly common, are very common in hospitalized patients, they're highly morbid, mortal often, and contribute substantially to the incidence and progression of chronic kidney disease. We've developed few new therapies because we don't have as good an understanding as we would like to have for what's actually happening in the kidney in individual patients. And so to improve the public health, the rationale for the study is we need these research level biopsies of the common progressive kidney diseases that cause the most harm and affect the lives of our patients living with kidney diseases. The key inclusion-exclusion criteria are on this slide. The inclusion criteria are simple. Kidney disease in participants with either type 1 or type 2 diabetes or so-called diabetic kidney disease, hypertensive kidney disease, and acute kidney injury. We exclude children, people with heavy proteinuria or known glomerular disease, transplants, malignancy, pregnancy, and increased biopsy complication risk. Now, after this study was initiated, it has become clear that as we're living in the midst of the COVID-19 pandemic, that unfortunately there are a lot of kidney manifestations of COVID-19 infection. Ill hospitalized patients with COVID-19 have very high rates of acute kidney injury. with tubular epithelial cell injury being quite common, as you see here in panel A. These are from a soon-to-be published multi-institutional survey of COVID-19 where kidney biopsies have been done for clinical purposes. We are seeing examples of thrombotic microangiopathy, as you see in panel E and F here, and then focal glomerulosclerosis in some individuals, and particularly collapsing glomerulopathy, as you see in panels B, C, and D. We felt it was our responsibility to address COVID-19 both from an acute kidney injury and a chronic kidney disease perspective, and so we have recently initiated a modular COVID-19 special population protocol within the KPMP, so that participants will have biosample collection, questionnaires, remnant tissues from clinically indicated kidney biopsies when hospitalized, and then will be asked to consent into the main longitudinal follow-up and kidney biopsy several months after discharge from the hospital. This study is designed from the get-go to have at least a 10-year timeline. We are currently here in phase 2, where we've established protocols, initiated the study, had more than 40 participants undergo kidney biopsies already in the study, and now we're enriching the kidney atlas and working on the next generation of tissue interrogation assays. The KPMP network or consortium has grown into a large network since the onset of the study 3 years ago. We now have over 40 different major academic medical centers in the US that are participating in this study, either as recruitment sites where participants are enrolled, consented, undergo kidney biopsies and longitudinal follow-up and biosample collection. We have so-called tissue interrogation sites where the kidney tissue from the biopsies are shipped to state-of-the-art omics type technology and kidney imaging, and you'll hear much more about this from Michael Eden in the subsequent talk. We have added over 18 different opportunity pool sites who bring richness, new richness via new technologies, clinical phenotyping, healthy reference tissue, aids to quality assurance for the study into the consortium over time. So this is really a very dynamic and growing consortium that allows KPMP to move ahead. The challenges that we face are how to work with the fire hose of data, the types of data that are already starting to come into KPMP. How do we integrate the clinical and phenotypic data with pathological data and molecular data? And the underpinning for this to be successful, how do we do sufficient quality assessment and quality control on the data using so-called metadata or data about the data collected rigorously so we understand what all of the data refers to? How do we curate the data to make sure that we understand and we have the right terminology to describe the data that we're seeing? And how does this all contribute to data analysis? So we have substantial efforts underway to make sure that we are robustly and rigorously quality controlling the data so it will be of optimal use within KPMP and to outside investigators as well. Part of that effort means going back to basics and developing a controlled vocabulary and standardized nomenclature that enables this data to be used in the best way. This also includes the development of more specific ontologies that allow the data to be computable in KPMP. And there's been extensive work that involves KPMP, led by KPMP, but now involves other consortium as well, that are such as HubMap or the Human Cell Atlas and other clinical consortia as well, to make sure we're all using the same vocabulary whenever we talk about the kinds of data that are being developed inside KPMP. We spend a lot of time thinking about the handoffs from the recruitment site to the biopsy specimens, sample handling, how that data gets to the tissue interrogation sites, or those samples get to the tissue interrogation sites. Data is generated, submitted into a data lake from which we create a knowledge environment through data transformation, data integration, and then data display in ways that are usable for a larger community. So a high-level view of this is we have participants that are recruited into the study, from which biosamples, clinical data, and biopsies are obtained. This goes to a central biorepository housed at the University of Michigan. These data are sent to the tissue interrogation sites, and data comes back to the central hub for the study for data integration and ultimate development into a kidney atlas. The kinds of data are multimodal and multi-parametric. So we've got genomic data, epigenomic data, transcriptomic data, microRNA data, proteomic, and spatially resolved metabolomic data, as well as the clinical information. And the kinds of data you can kind of scan in and scan out. You can get down to single cell level, looking at subcellular data as well, looking at cellular data, regional data, and then whole tissue display as well. Now, we've been, as mentioned, we've had about 40 biopsies so far in KPMP, and there are early suggestions from looking at these data that KPMP will be game-changing. It's still quite early days for this study, but we're starting to see convergence in the pathology between AKI and CKD, and in particular for the molecular pathways. So disease entities that we consider entirely separable may turn out not to be quite so separable on a molecular level. We're seeing discordance between the clinical adjudication as to what is the cause of the disease process and what's actually seen in the pathology. And in some cases, the biopsy findings are already changing management for individual patients. So this is early days, but already there's a lot of data coming into KPMP. It is our intention from the beginning that these data be a public research for a large community. And we want to make it easy for anyone to collaborate with the KPMP, putting data into the public domain. In some cases, if it contains a potentially identifiable information, then it's protected under a data use agreement. But the KPMP materials, the biosamples, and the data are designed to be used by a large community. And we want this to be highly collaborative and to provide access to these types of data. You can now go to our KPMP website, which here you can see the URL. As of today, we have 46 active patients, participants. We actually have 41 biopsies because on the date this is being recorded, a biopsy for KPMP took place, and over 500 data sets that are on file that are accessible on the KPMP website. It's easy to access. If you go on the website, hey, researchers looking for data, and we are continually building new tools to make these data more accessible to different kinds of users who would want to see the data. Our way of thinking about this is that there are different personas that we're creating this data for. Of course, there are researchers, there are data analysts, data miners, so-called power users of the data, or people who just want to see summary data, but also we want these data to be accessible for participants and for other patients to a certain degree with kidney diseases, to pathologists and to clinicians, so that it will be useful for the entire kidney community. Amongst the many precision medicine studies, I would say that KPMP is relatively unique in its involvement of the entire community, especially engaging patients in everything that we do as equitable partners. We really promote a culture of safety first for participants. We're addressing the big public health problems. Rare diseases are very important, but most most precision medicine projects don't necessarily address the big public health diseases like CKD and AKI that contribute to kidney failure. There's a clinical orientation to the Atlas in health and disease, and of course we're using next generation technologies with the use of machine learning as well. So I'll end by saying if you want to go fast, go alone. If you want to go far, go together. We believe that the way we're going to get to where we need to be to provide better treatments for our patients is to work collaboratively as a consortium and collaboratively with the larger kidney community. So that's the Kidney Precision Medicine Project in a nutshell. Let's find a breakthrough in kidney health together, and we look forward to hearing from Mike Eden and Laura Mariani, who provide more details on the kinds of data that are emerging now from KPMP. Thank you very much for your attention.